Hello, and welcome to Sciencell's first webinar in our series of webinars about qPCR. I'm Sciencell R&D scientist Jennifer Tung, and I'll be your guide today as we delineate how one achieves optimal sensitivity and specificity in qPCR experiments. We'll first go over the benefits of qPCR for those who aren't particularly familiar with this method, followed by an introduction to common problems faced by qPCR users. The crux of our presentation will then focus on how to prevent, detect, and address those problems. We'll finish with a brief introduction to our GeneQuery qPCR array products and end with an example of how qPCR data can be used to confirm, augment, or inspire future studies. Immediately following our presentation will be an extended Q&A session with members of the Sciencell R&D team to address any questions or concerns you may have on the subject. As a courtesy, please save your questions and comments for the Q&A at the end of the presentation. If you find yourself with a question regarding something specific in the presentation, please note your question as well as the relevant slide number located in the upper right corner of each slide so we can reference it. FYI, the slide number won't be so big on all the other slides. That's just so I can be reasonably sure you know what I'm talking about right now. Also, don't worry too much about taking notes because the presentation in its entirety will be available for download tomorrow. As a side note, we will be highlighting our GeneQuery endothelial cell biology array in determining endothelial genes affected by an abundance of VEGF-A during our presentation. If you find yourself interested in GeneQuery products, you can browse our products online at sciencellonline.com and navigate to the gene expression profiling section of our website. If you have the time, please take our survey at the end of the webinar to receive an extra 5% off your next GeneQuery order, in addition to whatever other promotional discounts we currently offer. Okay, let's get started. To exploit and troubleshoot qPCR most effectively, one has to understand it thoroughly. qPCR is simply quantitative PCR. The basic principles of it rely on the fact that PCR amplifies nucleic acid sequences in a predictable manner. The main difference is that in qPCR, a fluorescent reporter is integrated into new double-stranded DNA, and that fluorescence is quantified after each cycle. Popular uses for qPCR include, but are not limited to, determining the precise concentration of nucleic acid in your starting material, estimating gene copy numbers in a sample, and comparing relative gene expression. Before we progress, there are a few terms I'd like to define, since I'll be using them throughout the presentation. The first is amplification curve. Amplification curves represent the real-time fluorescence readings of your qPCR reactions after each replication cycle. The curve is divided into 3 phases. Initiation in yellow, which helps determine your background levels and threshold. The exponential phase in gray that signifies detectable target amplification. And the plateau in blue, which signifies the end of reliable target replication. The second is threshold. This actually won't come up again, but you need to know what it is to understand the next term. The threshold is basically any statistically significant point above baseline in the linear phase. The threshold is set to exclude background and ignore amplification outside of your primer's reliable detection limits, and you can essentially set it to be anywhere in your exponential phase. In this example here, I've set it rather high so you can see it. You'll need the threshold to determine the CQ, which is our second term. CQ stands for quantitation cycle, and it refers to the amplification cycle in which fluorescence exceeds background levels and is first detected. It is determined by calculating the intersection of your amplification curve with the threshold line. In short, it's the first cycle in which your qPCR experiment is able to detect amplified target product above background. So in general, the lower your CQ value is, the more gene expression you have. People commonly use it interchangeably with CT, or threshold cycle, but they are actually subtly different in terms of how they're calculated. Another term you might hear for CQ is CP, or crossing point. Using the CQ values for each reaction, you can calculate delta CQ, or the difference between your sample and your reference or control genes. This is used to normalize your data internally. From delta CQ, you can obtain CQ expression for each gene by exponentially transforming it, and taking the difference of the CQ expression between two samples gives you delta delta CQ, or the relative difference in gene expression between those two samples. Delta delta CQ expression is typically what researchers use to determine percent knockdown or fold changes in gene expression. Okay, moving on. So if qPCR is basically just fancy PCR, why do so many people laud it as the gold standard in gene expression analysis? Well, unlike other methods for gene profiling, qPCR is more targeted while still maintaining an exploratory nature. In qPCR, you can survey 1 to 384 genes concurrently, and you choose those genes based on targeted markers, functions, or diseases. It is true that other methods like RNA-seq and microarrays survey the entire transcriptome, which would include all of those things. But because of this, those methods sacrifice statistical power and can therefore yield ambiguous results that are difficult to interpret. With the less targeted, high-throughput methods, one can easily miss subtle but important changes in gene expression. Because of these setbacks, qPCR is frequently used to confirm exploratory high-throughput results anyway. In addition, qPCR is quick, flexible in its design, and a lot more affordable than other high-throughput methods. Aside from basic reagent and apparatus costs being less, the sample concentration requirements are much lower due to qPCR's sensitivity, so you save in that sense too. So why isn't everyone using qPCR? I do not know, but I suspect it's because designing and optimizing an array can seem a little intimidating. Because it's so sensitive, it seems easy to compromise your experiment unintentionally, and it actually is rather easy. But I'm going to take the time now to tell you about common qPCR errors so you can avoid, detect, and correct them in your future. One common source of error is in sample or reagent handling, which we call technical difficulties. Because qPCR quantifies things during the exponential phase, small inconsistencies introduced during sample pipetting or reagent handling will be amplified and quantified as well. As with any lab experiment, contamination is also a source of concern, and in this case, one needs to mine the ever-prevalent genomic DNA, or gDNA. Luckily, all of these hurdles are easily circumvented with good lab practice and attention to detail, and they can be easily detected with proper qPCR internal controls. Choosing the genes to include in your array design can also be a source of stress. Aside from the actual primers, which we'll talk about in a minute, you also have to have near expert knowledge on your subject so you don't miss any important aspects. Furthermore, for each gene, you have to decide whether you're interested in all splice variants or just specific ones. Again, this can be resolved with extensive literature research or consulting or collaborating with an expert. It might take a lot of time, but at least the solution is clear. Now, arguably, the most important part to designing an optimized qPCR experiment is designing and using good optimized primers, since qPCR sensitivity is only as good as its primer's efficiency. Aside from just being a little tedious, this aspect of qPCR design is also where unwanted and uncontrolled variability is most likely to be introduced. Because each gene in your qPCR array requires its own set of primers, careful attention has to be given to each primer pair and then to the set as a whole to ensure that all primers operate under the same conditions with the same efficiency. Otherwise, your data will be compromised. For example, quantifying gene expression of a gene that is inefficiently or variably amplified would be of little use, right? Especially if you're trying to compare its relative expression to a gene that is efficiently amplified. What's more, each failed or suboptimal primer pair represents reagents, potentially precious sample, and time gone. These issues are less easy to resolve, but I'll go over what we do at Sciencell to prevent, detect, and rectify these problems and ensure that our qPCR primers are fully optimized. At Sciencell, we pay particular attention to three main categories of factors that can affect primer function when designing and optimizing our primers, and those are consistency, dynamic range, and efficiency, precision, and accuracy, which I kind of lump together as one thing. Let's talk more about each of these. Consistency refers to having uniform parameters like primer annealing temperature, product size, primer size, GC content, etc. for all of the primers used in a single array. This is important because all qPCR primers will be subject to the same operating conditions, and the whole key to qPCR relies on the amplification step being fairly predictable, i.e. a doubling each cycle. If you have a primer pair with an optimal annealing temp 8 degrees off from all of the other primers in your array, that particular gene will likely get amplified differently compared to all of the other ones, and you might not even realize it. Maybe it only gets 1.5 doublings per cycle instead of 2. Likewise, if you have a primer set that produces an 800 base pair product instead of an 80 base pair one like all of your other primer pairs, then that 800 base pair product may not get amplified efficiently every cycle because it takes longer to replicate larger products. These differences may not seem like a big deal, but replications on an exponential scale would translate them into very big differences in your data. In designing primers, you also want to avoid primer dimers and hairpins, especially at the 3' annealing end. It's very important that you avoid primer dimers and hairpins, because if your primers are annealing to themselves or to each other, then they're not annealing to the target gene, which means they're also not amplifying your target gene, thereby decreasing efficiency. The specifications listed here are the ones that we at Sciencell have found most effective for designing primers under uniform conditions while still maintaining specificity for its target gene and amplification efficiency. We design all of our primers under these parameters so that you can compare results between different arrays or even mix and match genes from multiple arrays without having to worry about changing qPCR operating conditions. Our primers are also designed to detect all splice variants for its target gene using the KEGG database as our reference. Now, dynamic range refers to a primer set's detection limit. This isn't really something you can control, but it is something you should note because your quantifications will only be reliable within that detection limit. The standard method for determining a primer set's dynamic range is to use that primer set on known quantities of its target gene serially diluted. The CQ output is then plotted against the concentration on a log scale, and the linear portion of that plot will indicate the primer's dynamic range. Anything outside of that linear portion should be considered too high or too low for reliable quantification. For your reference, the typical dynamic range of a qPCR primer set is 10 to 10 to the 9th. Primer efficiency is sort of a catch-all term to refer to how well a primer pair functions in amplifying its target. Many factors contribute to primer efficiency, and we'll talk about some of those in a minute. But first, I'd like to address how you determine if your primers even are efficient, because many researchers don't even realize that they're using inefficient primers. The most stringent way to determine primer efficiency is to run your reaction using a known concentration of target and then plot it against a calibration curve. If you detect the amount you measured in your initial sample plus or minus 10%, then it's efficient. If you detect a lower amount, then your primers are not efficient and are likely not annealing optimally, or there is sample contamination. If you detect a higher amount, then your reaction may be contaminated with PCR inhibitors or non-specific products. A much faster and easier way to gauge amplification efficiency is by looking at the amplification curve generated by the qPCR reaction. This is what we do at Sciencell, and what's shown on this slide is actually an example of how we excluded two primer pairs from our GeneQuery arrays. For amplification curve analysis, you simply plot your amplification curves alongside a positive control. In this case, our positive control curve for gene 1 is a teal-blue color. Now you compare the slopes of the linear portions. They should be roughly parallel, and anything over or under that slope would be considered suboptimal. While this method for detecting amplification efficiency is quick, it can be tricky. For instance, if you're not paying attention, you might look at gene 2, highlighted in brown, and assume those primers are efficient enough because the slope is kind of close, and when you run the product on the gel, the band is very bright. However, when you overlay the control slope on that curve, you can see very clearly the slopes are different, indicating inefficiency. This means that the results for gene 2 are compromised despite its expression being quite high. To drive the point home further, I'd like to bring your attention to genes 3 and 4, highlighted in red and green, respectively. Both yield faint bands on a gel and have higher CQs, so at first you might be tempted to discard both of those as inefficient primers, or maybe you conclude that both of those genes just exist in low basal copy numbers. But analyzing the amplification curve slopes reveals that the red number 3 primers are actually inefficient, yielding unreliable results, while the green number 4 primers do work well and indicate actual low gene expression. Again, at Sciencell, we exclude primers that are inefficient or too efficient when designing our qPCR arrays to give lovely amplification curves like this one from our qPCR array for human angiogenesis. All right, now you can quickly assess primer efficiency. What can you do to affect it? Two factors that contribute to primer efficiency are primer precision and primer accuracy, or in essence, primer specificity. To determine if primers are precise, many researchers use melting curve analysis, which plots the negative derivative of the melting curve against temperature. In this analysis, each peak represents 50% dissociation of a double-stranded DNA product. Therefore, multiple peaks usually indicates multiple products, like gene number 1 in light blue. At Sciencell, we run our products through gel electrophoresis in addition to performing melting curve analysis because we know that there are always exceptions. For instance, by looking at these melting peaks, one might assume the dark blue and teal blue primers for genes 2 and 3 are precise and only amplify the target sequence. But running a gel for the products indicates that the dark blue gene 3 primers are also imprecise because they yield 2 bands even though there's only one melting peak. This can happen when the GC content of one product compensates for its size difference to another product in the same reaction. That might seem improbable, but this example is actually from our GeneQuery quality control testing, so it does happen. Now, primer accuracy refers to whether or not the amplified product is actually your target sequence. Amplification curve analysis is unlikely to reveal anything since things are still being amplified, and melting curve analysis can be ambiguous. As you can see from this example, there's clearly something up with gene 1 highlighted in gray, but what of the other two? Maybe something is wrong with gene 2 as well. That's not a very smooth peak, but you don't really know for sure. Since all of our primers are designed to produce a product sized between 80 and 230 base pairs, you can see only after running a gel that gene 2 in pink is also off target, as you might have suspected, while gene 3 in turquoise is fine. As I've shown you, the story for each primer pair is incomplete by looking at any one of these analyses alone, so we recommend that you do all three. Analyze the amplification curves, assess the melting peaks, and run the agarose gel. We also recommend that you avoid SNPs when designing your primers and to test your qPCR primers using a variety of cDNAs to ensure that your primers really are accurate and precise. We do all of these things at Sciencell because we know that reliable primers means a reliable and reproducible qPCR. We go to great lengths to optimize our primers so our qPCRs are sensitive enough to detect both high and low levels of gene expression while still having low standard deviations, as exemplified by this graph produced here in quality control on a mix of over 10 different cDNAs isolated from our own primary cell cultures. To drive the point home that the quality of your primers directly affects the quality of your qPCR, here are some more examples of what happens when you are not so diligent. The above 3 panels are just refreshers from the previous slides on how to detect primer inefficiency, imprecision, and inaccuracy. The bottom portion of this slide is data from 4 repeats of the same primers done for quality control. The first column on the left shows optimization data for primer set 1. It's great. The amplification curves are tight. The final fluorescent signal where the amplification curves plateau, highlighted in gray, are on point. The melting peaks are uniform, showing one solid peak, and the gel band is lovely. Primer set 2 in the middle column has some problems. The amplification curves are parallel but variable, and as a result of this, the final fluorescent signals are also variable. Furthermore, that uneven business there with the melting peaks seems to indicate there might be unspecific products, SNPs, or something else, even if the gel band doesn't indicate as much. From this, we can infer that primer set 2 probably has some issues with precision and possibly disagreeable reaction conditions, all leading to unreliable results. Primer set 3 from the right column again looks fine based on amplification curves, melting peaks, and its band. However, the final fluorescent signal is variable. This is likely the result of inconsistent pipetting during sample or reaction preparation, reinforcing the necessity of good lab habits. I'd like to bring your attention back to the middle column for primer set 2. If we look more closely at the amplification graph, you can just discern that there are minor differences in the CQ value, highlighted here in yellow. If you never looked at the melting peaks for this primer set, or if you neglected to do replicates, you would never know that these minor differences translate on an exponential scale to yield pretty big differences in final readings. But luckily, you now know what to look for when you're optimizing your qPCR primers. So great. But maybe all that seems like a lot of work, and you don't have that much time. That's okay, because there are companies like Sciencell that manufacture qPCR arrays and primers who will do all of that work for you. With manufactured primers, you get all the time and money you would have spent picking genes, designing primers, and optimizing everything back in your life so you can do other things. Our qPCR products are called GeneQuery and can be purchased as both individual primer sets and pre-designed arrays to survey specific functions, pathways, or diseases. GeneQuery primers have been tested and optimized according to all of the guidelines mentioned in this presentation. They recognize all splice variants for each gene, require only 100 picograms of starting material, and are very affordable. And gene selection for our pre-designed arrays are made based on notability through extensive literature research from high-impact articles. As an example, here's a depiction of our human endothelial cell biology array. The array comes with 88 genes relevant to endothelial cell biology and can be grouped according to cellular function, as shown here. Each of our arrays also comes with 8 control genes, so you can easily detect aberrancies in your qPCR. The first 5 control genes are housekeeping genes, so you can normalize your data. GDC is the genomic DNA control. It contains a primer set targeting a non-transcribed region of the genome to detect gDNA contamination. PPC is the positive PCR control that consists of DNA not known to be present in the human genome and a primer set that can amplify it. This control tests PCR efficiency and can detect the presence of PCR inhibitors. NTC is the no-template control that contains primers for beta-actin. You should leave this well devoid of sample to detect nucleic acid contamination introduced during general workflow. The array itself is pretty easy to use since we've done all of the optimizing for you. All you would have to do is add your template and reagents before running your qPCR. The way we've grouped these genes here is also just only one example of how those genes can be organized. For instance, the same genes for the same array can be grouped according to vessel function as shown here, rather than the cell biological functions on the previous slide. If you're unhappy with the balance of genes we included, for instance, maybe you'd like to focus more on angiogenesis and less on cell cycling, then you can work with one of our GeneQuery scientists directly to create an array that better fits your needs. But moving forward, let's say you now have your qPCR array with optimized primers. What can you do with it? Well, one of the most common applications of qPCR in basic science research is probably comparing relative gene expression between two cell populations. For example, you could take two identical plates of endothelial cells cultured in basal, serum-free endothelial cell medium and add VEGF-A, an angiogenic growth factor, to one of them. Performing qPCR on cells with and without VEGF would then reveal VEGF-induced changes in gene expression. We used our endothelial cell biology array that you just saw in just such an experiment, and we found several genes that were up or down-regulated as depicted in this graph of delta-delta CQ values from VEGF-treated cells relative to untreated cells. Since this is relative expression, the red line marks the reference point at 1. Anything above 1 is upregulated, and anything below 1 is downregulated. For our purposes, we only considered genes that were massively changed in expression, and those genes are listed in the upper right, highlighted in either red or blue. Red highlighting means we found those genes to be downregulated. Blue means up. The underlined bold genes are reported to be directly regulated by VEGF in some capacity, and the genes that are not underlined and bolded have no obvious or direct reported connection based on a quick PubMed search. As expected, there are a lot of angiogenesis genes that are influenced by VEGF, consistent with previous studies. Nothing unusual there. But what might be interesting is this column of shear stress and permeability genes, where only a few of them are reported to be directly regulated by VEGF. One might then wonder if there are more shear stress genes that can be regulated by VEGF in the absence of shear stress. Well, we have an array for that too, and thus a future study is born. Indeed, we offer many arrays on many different subjects, but here is a depiction of our endothelial shear stress and mechanotransduction array. It's the same basic principles as our other arrays with all the primers designed under the same conditions, so you could even mix and match genes from the two arrays if you so desired. Perhaps of even more interest is this column of unbolded inflammation genes that came up in the initial VEGF screen. But in this hypothetical scenario, you're an endothelial cell expert, and your knowledge of the inflammatory immune response is rudimentary at best. Plus, maybe you'd like to stay an endothelial cell expert and are only interested in inflammation as it pertains to the VEGF response. Well, that's fine too, because Sciencell conveniently offers individual gene primers in addition to the pre-designed arrays. Furthermore, they're already organized by cell biological functions, physiological functions, or diseases, so all you have to do is navigate to the inflammation category to see all of the gene primers we currently have for that category. Maybe while browsing our database, you'll decide that you're interested in how PRRs respond to VEGF2, and you can end up designing your own full array. Either way, you can order as few or as many of our optimized gene primers as you'd like. And if you're looking for a particular gene but can't find it, you can contact one of our GeneQuery scientists who will work with you to get you what you need. We end our presentation here, hopefully leaving qPCR more accessible to you and inspiring you to do your own qPCR arrays to discover new and interesting connections. Hopefully you've learned that the quality of your qPCR primers will directly affect the quality and reliability of your qPCR results. And you've also learned that in optimizing your primers by minding multiple parameters or buying them pre-optimized, you can optimize the sensitivity and specificity of your qPCR. If you'd like any more information about the products used and featured in this presentation, here's a list of them. Again, if you feel like you missed something, want to see something again, or just really enjoy the sound of my voice, this entire webinar will be available for download on our website starting tomorrow. As a reminder, we'll be offering an additional 5% off your next GeneQuery order to thank you for attending our webinar, and you'll receive that discount code after completing the survey at the end of this webinar. Thanks so much for listening, and we'll take any questions or comments you may have right now. And while you type in those questions, you might hear some noises shuffling around. That's me handing our microphone over to one of our other R&D scientists, Daniel, while I drink some water.
Hello, everyone. This is Daniel. I'm an R&D scientist at Sciencell. I'd like to take this chance to answer any questions you may have. Feel free to type in your questions or raise a hand to ask. During the talk, we already have some questions. One question is asking, do you have kits for mouse samples? And currently, all animal kits, currently we only have human kits, and all animal kits will be custom made at no extra charge though, which is $240 per 96-well plate or $45 for one primer set. The turnaround time will be 2 to 3 weeks for individual primer sets or 3 to 4 weeks for plates. Another question asked, what does the kit include? Well, we have lyophilized primer sets in a plate type of your choice, and we will include extra membrane seals for the plates and DPC treated water. The reagents that you should prepare are the cDNA samples and the cyber green dye or master mix of your choice. Okay, here's another question. What kind of samples can the arrays use? The array should use the complementary DNA samples that are transcribed from your total RNA samples. Okay, what differentiates GeneQuery from other qPCR products? Well, compared to other companies' similar products, in addition to pathways, biological pathways and diseases-related kits, we also offer kits focusing on human primary cell biology. I believe most of our audience here work with human primary cells. And other than that, our customized kits can better fit your needs. And for a limited time, there will be no extra charge for customization. And someone asks how to customize GeneQuery kits. There are multiple ways. If you have a specific set of genes that you to analyze, you can go to our website and upload that gene list directly as a PDF, Word, or Excel file. Or if you want to alter the pre-designed kit, simply send us email and one of our GeneQuery scientists will help you. Or if you want to select from our available gene primer sets, just go to our primer database website and you can select all the genes from our database. There's a button on your screen says raise a hand. If you want to ask a question directly in voice, you can click that button now. Okay, now I'm handing back the headset to Jenny.
Hey guys. All right, well, if there are no other questions, then we'll go ahead and conclude our webinar now. Again, if you have the time, please fill out the survey that pops up once we finish this and get your additional 5% off of your GeneQuery orders. If you have a question later, or if you can't figure out how to ask the question now, feel free to go online to sciencellonline.com and ask it there. One of our GeneQuery scientists will get back to you with an answer. Thanks so much again for attending. Bye-bye.